So I got into research really at quite a young age. I've always been around fish. I've always loved to fish. I remember as a young boy, 10 years old, loved to go out fishing. I remember going to our local store and buying this packaged fly rod because I wanted to learn how to fly fish when I was about 10 years old. And it was a Shakespeare rod combo, and I remember going on the street and started to practice and read how to fly cast. I became almost part of this kind of cryptic group of people in my neighborhood that I didn't even know knew how to fly fish. And my neighbors would stop, many of them that I didn't even talk to at the time, would stop and start giving me tips on how to fly fish. And so really from that time, I knew I wanted to be a marine biologist and wanted to do research. Bonefish are an iconic species here in the Bahamas. You don't have to go any further than looking at the 10-cent coin that depicts a bonefish on it. Here we have a recreational catch and release fishery that's worth $141 million to the local economy. And we know very little about the fundamental biology of this species.
So in 2015, the FLATS program, along with collaborators from various institutions around the US and Canada, started this project to study where bonefish on Eleuthera go to spawn. The study was designed to look at all of these different populations moving to spawning aggregations over the course of 3 years. So right now we're in year 2 of the project, where we've identified 6 suspected spawning sites around Eleuthera. So in year 1 of the study, we're using passive acoustic telemetry to determine where these fish are moving for their spawning aggregations. So we tag fish with transmitters that ping at unique rates. So whenever that fish swims past an underwater listening station, its transmitter, its unique ping, is coded and recorded by that underwater listening station. And in year 2 of the study, we took those narrowed down areas and we concentrated receivers around those areas. Each fish was implanted surgically with an acoustic transmitter, and those transmitters last 3 years, pinging once every 60 to 120 seconds. And they're pretty passive in the body of the fish. It doesn't interfere with their swimming patterns or anything like that.
So bonefish in the Bahamas has been heavily studied for the last decade, and this was really spearheaded through the Fisheries Conservation Foundation, led by researchers such as Dave Phillip, Andy Danielchuk, Steve Cook, Tony Goldberg, and Jeff Koppelman. And over the years, the research that they conducted was really heavily focused on things like the population genetics of bonefish and the best handling practices for anglers and catch and release anglers. And so the research from there really took a natural progression, and we started looking at what are the ecological roles that bonefish play in the ecosystem. And so we were interested in looking at predator-prey interactions between sharks and bonefish. From there, we've made collaborations with Florida Atlantic University and the Bonefish and Tarpon Trust and the Florida Institute of Technology, really looking at the blood work and sort of trying to identify really the spawning behavior and the triggers of spawning in bonefish. As an avid fly fisherman, I love going out catching bonefish for research, but we also do use seine nets. So sometimes for some of these studies, they do require catching large amounts of fish in a short amount of time. And as any bone fisherman knows, that can be difficult to do always on the fly. Once we capture bonefish, generally we move them in large coolers in the backs of trucks or on boats, and really as quick as possible, move them into the lab. Holding them in the lab, actually bonefish, I'm actually really surprised. They do really, really well in the lab. We've had our bonefish in here for about a month and a half now, and they began feeding within 3 days of arriving in our tanks here. We're currently raising them in the lab and actually taking bloods from them, which can be screened for hormones. The idea is over time, over a long period of time, we're hopefully going to try and get these peaks in the hormones moving up. And with that, we can sort of start to identify sort of what are the triggers of these hormone spikes and what time can we go in and actually try and strip spawn some of these fish. Bonefish is a huge industry in many parts of the world, and although the bonefish here in Eleuthera is a small fraction of that, really understanding how to protect these species and the interconnections between, say, the Florida fishery, the Cuba fishery, the Bahamas fishery of bonefish is really important in the survival of this iconic species. So I think that what keeps me driving forward with the Bonefish Project is just how little we know about this species. And really, we're already looking towards the future and understanding the early life stages of this bonefish species. So looking at the Leptocephalus larvae and basically the lost years of the juveniles. We know that the sort of small larval stages recruit on certain beaches here in Eleuthera, and we know that we catch the adults on the flats when we go fishing. A lot of work still needs to be done on these lost years.